Using the protocol, the biophysical effects of different optogenetic actuators on cardiomyocyte activity can be tested to aid in the development of optogenetic experiments in cardiac tissue and whole hearts.
By combining the patch clamp technique with sarcomere tracking and carbon fiber-assisted force measurements, we can study the effects of GTACR1 photoactivation on the electrics and mechanics of ventricular cardiomyocytes.
Optogenetic inhibition can potentially be used for optical defibrillation. GTACR1 is an optogenetic tool that enables the silencing of cardiac activity.
This technique is fully applicable to other research fields, such as smooth and single-cell muscle cell studies.
Although this protocol can't be used for high-throughput screening, it provides a means for the comprehensive analysis of cardiomyocyte electrical and mechanical function.
So the saying goes, a picture says more than 1,000 words. How much more information can we get across in a video? Some of our tools and techniques involved in single myocyte stretching and in probe preparation are highly intricate, and it is much easier to convey them in a video compared to a description.
After the blood has been washed out of the saline-perfused heart from a 9 to 10-week-old New Zealand white rabbit, switch the perfusate to a low-calcium, high-potassium solution and perfuse the heart for 2 more minutes after the heart stops beating. Perfuse the enzyme solution. Start recirculating the enzyme.